Thank you so much, Julia, for this wonderful introduction. And thanks to the ALPS team for the invitation to come here today and present some of the work that we've done on mechanisms of action of psychedelic substances. So, yeah, I'm really excited to discuss this topic with you today. As we've heard quite a few times already during this conference, psychedelics induce this altered state of consciousness. And we also know that these effects probably originate in the brain. And we also know that these effects, so the biological effects, the subjective effects, have an impact on psychiatric patients. That has been shown in quite a few randomized clinical trials now. We've already discussed some of the limitations of these trials, but the evidence is definitely pointing there. And we're currently conducting some clinical trials also in Zurich, but this is not what I want to talk about today. I want to talk about what exactly it is that these substances do in the brain to have these effects, to find out what the mechanism of action is that eventually helps patients to get better. And the reason why I believe it is important to understand the mechanism of action is that understanding the mechanism of action will help us to optimize the therapeutic approach, to find the best therapy for our patients. And this is exactly what we should be aiming at in this field, to provide the best psychedelic-assisted therapy possible. And I believe that this is not possible if we don't care about the neurobiology that is giving rise to these beneficial effects. Now, when it comes to possible mechanisms of action, I'm sure that a lot of you in the audience probably have already an idea of why these substances could be beneficial in clinical populations. What some of you may not know is that we actually have no clue. We just don't know, right? There are, however, a lot of hypotheses, and some of these hypotheses have, stronger evidence than others. So let me just run a few of them by you. It's not a comprehensive list, but some which, at least sound reasonable to me. So there could be changes, long-term changes in the serotonin to A receptor system. As you've heard before, these drugs target the serotonin to A receptor system. So it's conceivable that there might be long-term changes in this receptor system. There might be induced neuroplasticity. I'm pretty sure that all of you have probably heard about that by now. And for this hypothesis, we indeed do have evidence, but unfortunately, so far only in animals. And we have no idea whether that translates to effects in humans, and whether that, even if it translates to effects in humans, whether that actually has something to do with these therapeutic effects. There could be an impact of psychedelics on neuroinflammation. There could be an impact on epigenetics. There could be changes, long-term changes in how our brain is organized. There could be changes in reward or emotion processing. I think some of this was already discussed today and yesterday. Another topic that has been discussed at least partially is increased insight in potentially dysfunctional behavior and increased social connectedness. So again, this is not a comprehensive list, but some hypotheses that have some data underlying which are conceivable that they may help patients. Now, I could spend at least 30 minutes talking on each and every of those topics. We don't have that time today, unfortunately, so I will have to focus on 3 of these topics, which is alterations in brain network, alterations in reward and emotion processing, and increased social connectedness. And I don't want to just stop there and just tell you about the evidence that we have so far that these mechanisms might be helpful. I also want to discuss how that translates into therapeutic approaches, or how we could think about the psychotherapy that is part of the psychedelic-assisted therapy, and what does that mean for being a therapist and how the therapist conducts the work. So let's start at the beginning. So we know that psychedelics target the serotonin 2A receptor. The serotonin 2A receptor is an excitatory receptor, and that means that it increases the chance that our neurons in the brain are becoming active, that they fire. Now, the serotonin 2A receptor is distributed across the whole brain. However, there are differences in density of this receptor depending on the brain region. So it is again conceivable that there are other changes in brain regions where there are a lot of these receptors versus in areas where there are fewer of these receptors. Of course, these changes in firing in the brain are also dependent on the dose that you administer. And they're dependent on the exact substance that is administered. I've pointed out the serotonin 2A receptor. Of course, this is not the only the single receptor that is being targeted. We do think it's the main target of these substances, but every single one of these different substances has a specific pharmacology and also targets other receptors that may modulate the effects. Now, what does that mean? Our cells fire different. Well, if our cells in our brain fire different, that has an impact on how the brain itself is processing information. Because if our cells, the brain is all about passing information from one region to the other to further analyze the information, to integrate the information. If our cells fire differently, that means that the way information is processed in our brain is different. And then in a last step, if our brain is processing information differently, that obviously has an impact on how we perceive our body and how we perceive our environment, because the brain is not an island. So, and I will walk you through some examples on each of these levels on what psychedelics do here. Let's start with information processing in the brain in general. So I know that not all of you in the audience are neuroscientists, so I'm giving you a very, very brief introduction into neuroanatomy. There will be quite a few pictures of brains showing up in the next couple of slides. Please bear with me. You don't need to know everything and every single detail about these areas. The only thing that you really need to know when interpreting these next couple of slides is that there is a difference between these gray areas here and the colored areas here. And the main difference is that the colored areas that you see here in the back of our brain, here what we call the temporal cortex, and up here, these brain areas are responsible for receiving input from our senses. You can see the colors down here: auditory, motor, touch, visual. So these areas receive input from the environment and from within ourselves, of course, too. While the gray areas here, they're responsible of making sense of this information. They're responsible of connecting this information, integrating this information, and connecting it with our memories, connecting it with our future planning, and enabling behavior so we can react upon whatever information we receive. So when we started out to investigate what these substances do in the brain, the first thing that we wanted to test is an hypothesis that was put forward by Franz Vollenweider and his colleague Mark Geyer. Based on animal research, they suggested that the thalamus, which is this structure here in the middle, is not able to filter the information like it usually does. So, of course, if we would just perceive everything in our environment, that would be too much. We could not really make sense of this information. That's why we have this thalamus, which is, is filtering information which is not important or where we're not currently paying attention to. And they suggested that under the influence of a psychedelic, the thalamus is not able to do this filtering anymore, and therefore a lot more information is passed through to the cortex, leading to a sensory overload, and with that causing these psychedelic effects. So, of course, what we did is we looked at how the thalamus is communicating with the rest of the brain. And we used a technique which is called seed-based connectivity, which is giving us the connectivity between the thalamus and every other region in the brain. And what you can see here, and now keep in mind the picture I showed in the beginning, right? The colored and the gray areas. And if you recall this picture, what you can see here is that the warm colors here overlap really nicely with these colored areas that we've seen before. And the gray areas overlap really nicely with these blue areas. So, what does that mean? That means that under the influence of, in this case, LSD, our thalamus is more strongly connected to our sensory brain regions, so the warm colors. Sensory brain regions is more connectivity. So closer communication to the sensory brain regions, but less communication to these associative brain regions, which are supposed to integrate the sensory information. So, so far so good. That makes sense. That is very much in line with what Franz Vollenweider and Mark Geyer proposed, right? The downside, the disadvantage of this method is that it does not, it's a correlation. It's not able to tell us anything about the direction of the connectivity. So that means that while the thalamus could be sending more information to these yellow and red brain areas, it could also be the opposite. So these areas could be sending more information to the thalamus. We just don't know. So we had to use a second method, which is called dynamic causal modeling. As you can see, all these techniques that we use in neuroimaging come with their advantages and disadvantages. The disadvantage with this method is that, unfortunately, for the time being, we cannot look at the whole brain. We have to pick specific regions which we are interested in, and we picked these specific regions. This is, of course, the thalamus and a few other cortical brain areas, and then we looked at the directed connectivity. Again, red colors increased connectivity, blue colors decreased connectivity. It's the same data set, it's still LSD. And what we do see here is that there is indeed increased connectivity from the thalamus to this cortical area here, the posterior cingulate cortex, decreased connectivity to some other cortical brain areas. So, so the thalamic filtering is different from what we from how the thalamus is usually performing in our regular waking state. But just a reminder again, same picture as before, keep it in mind, but we don't want to just stop by looking at the thalamus and its connectivity to the cortex, right? We also want to know what's going on in the cortex. How is information passed from one brain area to the other? And we did that with LSD and psilocybin, again, using a slightly different neuroimaging method, which is called global brain connectivity. So what we see here is how every voxel, so every little part of the brain, is connected to the whole rest of the brain. And what you can see here really nicely in both LSD and psilocybin is that you again get these two different networks, right? You see these yellow and red brain regions, again, heightened connectivity, which work very closely together. And these strongly overlap with our sensory brain regions. And again, these blue brain regions overlap very strongly with our associative brain regions. So, what does that mean? That means that we are in a state of heightened sensory processing, indicated by this strong connectivity between sensory brain regions, but our associative brain regions, they are disintegrated, meaning that the way we integrate this information, how we connect it to our current situation, to our past, to our future, etc., that is probably very different under the influence of a psychedelic, in this case here LSD and psilocybin. So to sum that up real quickly, we have seen increased filtering. Here the red dot is the thalamus. Increased or decreased thalamic filtering with heightened connectivity to our sensory brain regions, heightened connectivity between sensory brain regions, which is not counterbalanced by our ability to make associations, to integrate this information. And why do we think that this could be helpful for a patient? Well, if I am integrating the information, and again, this information can come from within me, but it can, of course, also come from our environment. So if the way I'm integrating this information is different, this may help me to break free from rigid thinking patterns, because I might be able to see myself in a new light. I might be able to perceive the world differently, and that may eventually help me to find new and better solutions to problems that I've been struggling with. Before we get to the therapeutic part, I want to give you a few more details on these resting state brain analysis, because I think, again, that they may be important for what exactly we do when it comes to psychedelic-assisted therapy. So this is psilocybin here. We have not only scanned our participants while they experienced the peak effects, we have scanned them 20 minutes after administration, we have scanned them 40 minutes after administration, and we have scanned them 70 minutes after administration. And what you can see here aligns really nicely with what our participants are telling us. So you see the first changes that appear in the brain are in our occipital cortex. And if you remember from the previous slides, the back of our brain, the occipital cortex, is responsible for receiving visual input. And this is usually what our participants tell us first, that the first thing they notice is that the floor or the walls start moving a little bit. And this usually happens around 20 to 30 minutes after administration. And then we see that 40 minutes, we see again the effect on the visual cortex is more pronounced, but additionally we see that this disintegration of our associative brain regions is already starting to happen after 40 minutes until we see the full-blown pattern during peak effects. Now, what I want you to keep in mind here is that, especially this picture here, because here participants don't quite experience the full peak effects yet, right? And theoretically, this could also be a proxy for just a lower dose, right? Because it hasn't quite unfolded yet. It is probably very similar to just a lower, not a minimal dose, but a lower dose of psilocybin. And remember that here, with this, after 40 minutes or potentially also with a lower dose, you already see this disintegration of information processing. So when it comes to therapeutic effects, of course, we want these effects to last, right? If people are just feeling better during these 60 minutes, during the 6 hours of acute effects, that's nice, but it's not really, really helpful. So we want these effects to last. So what does that mean when we're looking at brain networks over time? And unfortunately, there are, at this point, very, very few studies out there which investigated how these changes in connectivity change after the administration of a psychedelic. So this is one out of, I think, 2 studies which have looked at that. And as you can see here, we do see changes in these connectivity patterns even a month after administration, so 1 week, 1 month. it does seem that the pattern seems to change a little bit. So the effects that we are seeing acutely or even after 1 week are not necessarily the same that we see 1 month later, which obviously raises the question of how relevant they are. But on the other hand, obviously, we don't want to keep people in a brain state where they are experiencing psychedelic effects, right? So we don't necessarily expect that we just see a prolonged pattern of effects that is similar to the acute state. So while this raises a lot of questions, it still shows us that there is something going on, that the functional architecture of the brain is changed after the administration of a psychedelic. And the other result out of these studies that I wanted to show is this one. And what we see here is, or what we did here is, we looked at how our brain is organized when it's not under the influence of a psychedelic, so in the placebo condition. And we correlated this, the way it's organized under placebo, with the magnitude of changes induced by the psychedelic. And you can see here that there is a remarkably strong correlation. And what that means is that, I mean, we know that there's a massive inter-individual variability. So the way I experience a psychedelic might be totally different from the way you experience a psychedelic. And what this tells us is that this may have something to do with how our brain is organized on an individual level. So because my brain is slightly differently organized than your brain, you might have a different reaction to a psychedelic. And the interesting part here, when we relate this back to psychedelic-assisted therapy, is that, well, maybe we could measure the brain connectivity, the organization of a brain, not under the influence of a psychedelic, before we administer it, and maybe this tells us something about how this person might react to the psychedelic. So maybe we have a predictive biomarker. Again, this needs to be tested in clinical trials, as we're doing right now, but maybe we do have a predictive biomarker, and that would be a first in psychiatry. So, but here it might be. So I want to take this part of the talk full circle by coming back to the receptor level. We talked about neurons and firing and the serotonin to A receptor. So in the study we conducted with LSD, we did not just use LSD, but we also used a second substance which is called catanzerin. Now, catanzerin blocks the serotonin to A receptor. So basically, our participants come in, we administer catanzerin, we wait for an hour so that that catanzerin can actually work. At that time, the serotonin to A receptors of our participants are blocked. Then we administer LSD, and that means that LSD can then target all these receptors that it usually targets, except for the serotonin to A receptor, because that one is already blocked. That one cannot get activated. And then we compare that between LSD and placebo and LSD and catanzerin plus LSD. So that's the picture you've already seen before. And this picture, as you can see, we don't even need statistics to talk about that. What you can see here is that this looks remarkably similar. And the reason why I'm not just showing you catanzerin plus LSD versus placebo is because the picture is terribly boring. It is really just a gray brain. So meaning that once we block the serotonin to A receptor with 100 microgram of LSD, we reduce all subjective as well as these neuronal effects to a level where we cannot distinguish it and the participants cannot distinguish it from placebo. So the serotonin to A receptor really seems to be necessary for LSD to have the psychedelic effects it has. And we also, I mean, this is maybe a little bit, so this is probably interesting for the neuroscientists in the room. So we also used another method where we looked at receptor gene expression. So Where is the serotonin to A receptor located in the brain? We correlated that with these changes in connectivity that we saw under the influence of LSD. And again, we don't even really need statistics here. It's so obvious. You can see that the changes in connectivity are very much related to where the serotonin to A receptor is expressed in the brain. And now finally we get to what does this all mean for psychedelic-assisted therapy. And unfortunately, I will have more questions than answers, but I think these questions are important and we should try to answer them in future studies. So first of all, it seems like if that is true, and if these changes in the functional organization of the brain really have something to do with whether we get better or not when undergoing psychedelic-assisted therapy, that means that, well, these findings of new perspectives, this breaking free from the rigid thinking patterns, that this may be a key component of the therapy, and the therapist probably should actively support this. Now, as I said, there are more questions than answers here, because what exactly is then the best timing or dose to leverage this breakdown of rigid thinking patterns? We have seen that with even after 40 minutes already, we see this disintegration of our associative brain networks, which may give rise to this breakdown of rigid thinking patterns. So maybe that tells us that we don't even need such a high dose to be able to get to this effect. The other question is the timing, right? Usually if we are experiencing peak effects, that's not necessarily the time where our participants will want to talk about their problems, right? Usually you will start talking and working on the experience afterwards. So yeah, what is the best timing to do that? And of course, we don't know how long this pattern lasts. Right now, we just don't have these long-term studies. I mean, it might still be present the day after. It might be present for a week. We just don't know. Right now, we only know that it's present under the acute influence. Does that mean that maybe we should, encourage the participant to really think about new perspectives, to talk about new perspectives, even under the acute experience, under the acute influence? The next question is, well, is this acute experience, given that we see this pattern here, is that more important than these long-term changes in brain connectivity? And this relates to a certain degree back to the question, like, do we need the conscious experience of the psychedelic effects for them to help patients, or is it just a very biological matter here? And the other thing, which is, well, still a question, of course, but also a hope, maybe with psychedelics we'd be the first in psychiatry to actually have a stratifying biomarker that can tell us whether a patient will respond well to the treatment or not. Okay, so with that, we're switching gears a little bit and stop just looking at the brain as an entity, but start looking at the brain in interaction with the body and the environment. So in Zurich, we have run quite a few studies, and we have shown one thing repeatedly, and this is that under the influence of a psychedelic, our participants react less to negative stimuli. This is a behavioral task where you can see people, it's harder for participants to identify negative emotions, whereas there's no change in neutral or positive emotions. We've also done something similar in the scanner, and here we see that our emotional center of the brain, the amygdala, is less reactive to negative stimuli. Again, less processing of negative information. So one important way of how psychedelics, at least acutely, change our interaction with emotional environment and emotion processing in general. This is a study which is under the acute influence of a microdose. This is not exactly the same paradigm so here we did not show participants any negative images, but what we've done here is again resting state. But it is still interesting in terms of emotion processing, because we looked at the connectivity again between the amygdala and the rest of the brain. And what we saw under the influence of a microdose is that there are changes in connectivity between the amygdala and the rest of the cortex, which is what you can see here. Again, increased connectivity, decreased connectivity. But one thing in particular was very interesting here, and this is the connectivity between the amygdala and these frontal brain areas. And you can think about these frontal brain areas here as kind of the controlling instances of our emotional system. And here there's a stronger connection between the amygdala and these controlled brain regions. And the strength of this connectivity is also correlated with changes in positive mood. So even a low dose of psychedelics could change how we process emotion. Of course, we also know that most of these, or basically all of these microdosing studies, have not shown any behavioral effects. So people are usually not better at the tests that they complete under the influence of a microdose. But given that there are these subtle changes in how the brain is organized, even under the influence of a microdose, this may point to a potential therapeutic effect, because it might mean that while you don't, for people who don't have any problems with emotion regulation, this might just not have an impact. But it might theoretically help people who do have problems with emotion regulation, which is the case, for example, in depressed individuals. But again, this needs to be tested. This is all collected in healthy participants. So again, with this type of therapy, we want to see long-term effects. We don't want this to be a chronic treatment. It needs to have long-term effects. And again, another research group has looked at long-term changes in amygdala reactivity. So exactly what I showed you in the beginning, negative stimuli and the amygdala is less reactive. And what you can see here is, again, 1 day, 1 week, 1 month after psilocybin. And here you can see really nicely how we have this reduced amygdala reactivity 1 week after administration, and then it returns to baseline or to what it was like before 1 month after administration. And again, this could be really, really important, because we know that depressed patients have a negative bias. They react more strongly to negative information. And having this long-term effect on amygdala reactivity may point to a lasting effect on how we process, or how the patients process, negative information. But of course, in science, there's always a but, and that's another study, and this is actually in depressed patients after psilocybin. And what you can see here is exactly the opposite. So here we see increased amygdala reactivity the day after psilocybin administration. However, with those data, it needs to be noted that these data were collected before any integration therapy was conducted. So basically, right, or more or less, or the morning after psilocybin, which might represent a different state than 1 week after, for example. So again, what does that mean? So it seems like psychedelics may provide a window, an opportunity for processing negative life events. If our emotional system is less reactive to negative information, then maybe this is the time where we want to approach these negative life events that maybe also cause the depression or PTSD or whatever, or addiction. I mean, it's really a transdiagnostic issue. But again, so many questions. So when should we really confront patients with negative life events, or at least encourage them to process these negative life events, or the negative negative things that may have an impact on their illness? We've seen that in at least here it lasts up to 1 week, but of course we haven't measured, right? It could be up to 2 weeks, it could be 3 weeks. We just don't know. The only thing we know is that it seems, at least in healthy individuals, it returns to baseline at 4 weeks after administration. So, but what about this then, right? So we know that there is increased amygdala reactivity the morning after, which probably means that the morning after our participants, our patients, are in a pretty vulnerable state, in a very emotional state. So that kind of points to that the integration therapy is really necessary to achieve long-term improvements. But it also means that maybe we need to provide additional support in this post-acute phase. And again, long-term effects in clinical populations are currently absolutely unclear. These are the only long-term data we have so far, which is the morning after. But I promise you, in probably a few months, we will know how this picture looks in depressed individuals after a few weeks. But we need to wait a little bit longer until we have those results. So the last topic I wanted to talk about today is social cognition. So another very, very important factor in our environment and how our brain interacts with the environment. So we have seen this alterations in emotion processing. We've seen reduced reactivity towards negative stimuli. However, what does that mean for social stimuli, for social interaction? We know very well that our participants, or that depressed individuals, psychiatric patients in general, experience a lot of social rejection, a lot of exclusion from their social environment. So what do psychedelics do when it comes to negative social interaction? Well, so to test that, we used a task in the scanner again, which is called Cyberball, a very standard paradigm in psychology, actually. And the way it works is that we introduce our participant to 2 other people. In this case, it's Michael and Sarah. And we tell them that once the participant is in the scanner, they can play a game with them. And the game consists of the participants being able to throw a ball to either Michael or Sarah, and of course Michael and Sarah can do the same. Now what happens after a while is that Michael and Sarah decide not to throw the ball to our participant anymore, so they are excluding him or her from this game. And while that doesn't sound like the most terrible situation you've ever been in your life, it is pretty impressive. So basically every participant comes out of the scanner, at least in the placebo condition, and it's like, oh my God, that was really, really mean. So it does hurt, even though it might sound a little bit silly, but it is a very powerful paradigm. And the other reason why I really like this paradigm is that, first of all, it has been replicated many times. And what has been replicated is, while you're being excluded from this game, your anterior cingulate cortex lights up, so it's more active. And so this anterior cingulate cortex signal has been termed the social pain signal. So while you are experiencing social exclusion or social pain, this is what happens in your brain, so stronger activity here. And of course, as I said, it's a very standard paradigm. So other people have done that with various patient groups. For example, borderline personality disorder patients here. And here we see increased reactivity, increased social pain signal in this brain area. So what happens under psilocybin? Well, first of all, of course, we need to ask our participants how they felt, right? And we ask them a lot of questions because after all, I mean, they are under the influence of a moderate dose of a psychedelic. So first of all, we need to make sure that they, were even aware of what was happening. And I can promise you they were. So we asked them, for example, to guess how many times they have received the ball. And they were perfectly able to do that. They were perfectly aware that they were being excluded from this game. But when we asked, like, yeah, how strongly did you feel excluded? Then that was significantly reduced. So the feeling of being excluded was less in the psilocybin condition. And also when we look at the brain, we see that the social brain signal, the social pain signal in the anterior cingulate cortex is reduced under the influence of a psychedelic, and therefore, providing the opportunity of maybe normalizing this increased reactivity that we have seen in patient populations. We also did another paradigm on empathy. And what we've seen when we did this task under the influence of psilocybin is we see that participants show increased emotional empathy, so they feel more strongly with other people. We also looked at cognitive empathy. So cognitive empathy means, can you identify emotions in another person? That was not changed, so maybe this is just not something that you can acutely change with a psychedelic. But in a way, we were also very happy to see that result because it also was not, less cognitive empathy, so they did not get worse, which is always good for us because that means that our participants actually completed the task like they were instructed. They were not distracted by anything. So we can assume that this increase in emotional empathy is true. We also did a moral decision-making task, a moral dilemmas task. We did not find any changes there either. So at least during the acute phase of the effects, we don't find an impact with that dose on moral decision-making styles. And I think last result I want to show in this context is we also looked at how suggestible people are under the influence of a psychedelic, in this case LSD. And what we see here is we had two different conditions, low conflict and high conflict. And usually people tend to agree or tend to not agree, tend to change their mind about something when there is a high conflict, as you can see in the placebo condition. So low change of low adaptation to the other opinion if there's a low conflict, but high adaptation if there's a high conflict. So with a high conflict, the opinion of other people seems to have more of an effect, making you overthink your own opinion and then changing yours. And we see the exact opposite pattern under the influence of LSD. So with LSD, people agree or change their opinion more strongly when their own opinion is rather similar to someone else's. And this is also reflected when we look at activity in their brain. And again, obviously, this has therapeutic implications. So it seems to be difficult to confront someone, or maybe not advisable to confront someone with opinions that are very far from their own, because it seems like people do not really process them in a way that changes their decision-making behavior. Okay, implications for psychedelic-assisted therapy. So it seems like psychedelics provide a window of opportunity, provide for reconnecting with our social environment. I just picked a few of the social effects that we have seen under the influence of psychedelics. If you're more interested in learning more about social effects of psychedelics, we've written a review paper where we summarized all the literature to date. But yeah, so there seems to be an opportunity for reconnecting, and that may be especially important in psychiatric patients where we know that social withdrawal is a major characteristic of almost all psychiatric disorders. This may, of course, also have an impact on the therapist-patient relationship. And if you're thinking about the suggestibility results, for example, that obviously means that we need to be very responsible with the patient while they are under the influence of a psychedelic. But we could also speculate about group sessions, right? If we know that these substances have a positive impact on how we perceive our social environment, maybe we should create a social environment for these therapeutic sessions. The other important question, I think, is, well, what about family members? What about loved ones? Maybe it's worthwhile including them to a certain degree into the therapeutic process if the way they may perceive their social environment is changed. Again, almost no long-term data, so we need more studies to see if that effect is enduring or if that is really just something that happens acutely. And with that, I want to wrap it up. Just a few summary thoughts. So, psychedelic-assisted therapy is and will always be an interaction between a pharmacological and a non-pharmacological mechanism. So as we've seen, the brain, even if it's a really purely biological mechanism, the brain itself is not an island. The brain interacts with its environment, and there are pharmacological and non-pharmacological factors, and we just need to find out which are the important ones that contribute to the clinical effects. We've seen that maybe it's worthwhile to think about how therapists can support finding new perspective. We've seen that there might be an opportunity for processing negative life events. We need to figure out the best way of how to integrate this into the therapeutic process. We see this changes in social cognition that might be helpful, and we need to figure out how we can best leverage this effect. We might even have objective stratifying biomarkers, which, as I said already, is really cool and would be a first in psychiatry. And I think we should really take these neurobiological effects serious. We should take these questions serious. We should try to answer them as soon and as best as we can to reach this aim, I think, that we all share, to provide patients with the best psychedelic-assisted therapy possible. And with that, I want to thank you for your attention, and of course to all the people who have been involved in these studies that I just presented. Thank you so much. Yeah, and
no, please.
So I don't know how much time we have left, but if there are any questions, I'm of course very happy to answer them.
I was thinking we had about 15 minutes of questions each time, no?
Normally, yes, but
5 to 10
minutes.
All right, well, there's somebody right here.
Thank you. Thank you very much for your presentation. That was really interesting. I was asking myself, we know that intestines, the bowels, are our second brain. And I was asking myself, do we have any knowledge about what interaction there is with psychedelic to the intestinal bowels?
Yeah.
And study about this.
It's such a wonderful and interesting question, and I think it is especially important given that we know that there are a lot of serotonin to A receptors in our bowels. Yeah, I think it's worthwhile studying that. I think it would be really, really interesting, but for the time being, we don't know, at least not to my knowledge. I don't think that anyone has done a study on that yet. But we should put that on the list of potential hypotheses, and we should try to find out.
Okay. Hi. Hi. I have a question about when you spoke of the results with the microdosing and how you found even with the microdosing that there were changes in connectivity between the amygdala and the cortex. Are there comparable? Is there comparable data to antidepressants? And if antidepressants, what antidepressants do with that relationship?
So
Thanks.
we know that with antidepressants, so I'm only aware of these reactivity studies. So we know that antidepressants too change amygdala reactivity. But of course, the difference there is that it takes much longer. So you need to take the antidepressant chronically and for a long time. But then it has an impact on amygdala reactivity as well.
Hello, over here. So I wanted to ask you, so first I find really interesting that you have sort of a predictive way of understanding who can take the therapy in a positive way. So by looking at the activity beforehand. So I was also wondering if you have looked at the emotional or social processing of individuals before the therapy and do the same correlation, and somehow how would that be important also in deciding which individuals are prone to take the therapy?
Yes, absolutely. So the short answer is no, we don't have those data yet. But as I mentioned, and also with the functional connectivity, right, these results still need to be translated into a clinical population. So we need, it's a hypothesis right now. It's a promising hypothesis, but still a hypothesis at this point. But as I said, like, we will get the data of our depression trial sometime very soon now, and this is exactly where we did that, and this is exactly where we can look at these effects, and then hopefully, even though I'm not able to answer right now, hopefully if we talk again in half a year, I will be able to give you some answers to that.
I also have a question. I'm on the other side. Hi. Thank you very much for the very interesting talk. I just wanted, more than a question, maybe bring a point of discussion to the table, because in one of the publications that you showed, you looked at effective connectivity and used dynamic causal modeling specifically. And you mentioned that it wasn't possible to do DCM in whole brains. I just wanted to bring to your attention that there is a new model out there called regression DCM where you can now do that, but it has a few simplifications. So I'm not sure whether it would be good for your design or not, but I was wondering whether you've considered that.
Yes.
Okay.
We have, as you said, there are certain limitations right now in this model. There will be another one, and we're currently looking at these data with network DCMs. So this is giving you a little bit more of the full brain flavor than the tiny regions we have right now. So yeah, there will be data out there on network DCM sometime soon, and of course the DCM method is being developed further and further. So I'm sure we will have full brain DCMs that are not based on regression DCMs sometime soon, and I'm looking forward to applying them to the data. As I said, the first step is now the network DCM. It's not published yet, but it will be presented within the next few months.
Hi, hello, here in the back. Thank you for your presentation. I was wondering, because you raised the issue of the potential effect of group therapy and how it would help. So I was reading last week that some veterans suffering from PTSD, but also some people that had done ayahuasca ceremonies in the Amazon, both commented that having a group with whom to discuss afterwards actually helped even further their integration process. So I was wondering if you've had heard of this and if you had already tried group therapy and what were the results or views you had on this further?
Yeah. So we have, in our clinical trials, we have not done group therapy. As you might have seen in the movie that was screened yesterday, the study that Franz Vollenweider has led with the Zen Buddhists on Mount Rigi, that was a group administration. There are more and more studies now testing group administrations. I cannot, at this point, we just don't have the data yet, right? So I cannot tell you whether it's indeed better. I think there is evidence that points into this direction, that it could indeed have benefits, but we don't know yet. And of course, the way we design that, we need to be careful, because group dynamics can, they have their very own dynamics, which could be, positive, which could be negative. You still need to be able to take care of one participant specifically if they are going through, a rough time, for example, and they may not want to be in the group at this point. So there are, I think, a lot of things that we need to consider when thinking about group therapy. But I think it is worthwhile the effort of thinking about it and testing it and see. And maybe there are, again, in the end, always a little bit more complicated than you thought in the beginning. There might be people who benefit from a group session, and there might others who benefit from a single patient setting. But I do think that there might indeed be people who will do well with a group therapy session. So I think it's a way to take this work further and something that we should investigate and consider in the future.
Hi, over here front row. Yes, hello. I couldn't help but notice Carl Fristen's name up there, and I have less of a question than sort of an incentive for you to tell us how you think maybe psychedelics, what insight they give us into a greater model of cognition. A lot of people talk about relaxed priors being the key to how would you describe what your work sort of contributes in that context?
Yeah. So I mean, the relaxed priors model is I think it's a very good model that provides, at least to a certain degree, testable hypotheses. And this is great, right? Because at one point we want to start understanding what exactly it is that they do. But of course, as models tend to be, they're somewhat reductionist, right? They will not tell us, or they will not provide the full complexity of these effects. I think that the data that I showed speak to some of the effects of what is called the rebus model. For example, as we talked about, the decreased thalamic filtering, right? So more bottom-up information flow. That is basically what is also being described in this model, just in other words, in words of bottom-up versus top-down. The other thing that speaks to the rebus model in this data is this disintegration of associative brain regions, of higher order brain regions. Here I disagree to a certain degree with the model, because what we see is that it's not just the default mode network, but it's actually all associative cortices, including the default mode network. We haven't seen anything special about the default mode network in our data. But it is obviously, it's part of these associative cortices. So so the disintegration of these cortices could be an indication of relaxed priors, but it's a very indirect evidence of this. So we would need to test this model a little bit deeper to be able to verify or falsify it, but there are definitely overlapping aspects, and some of these results speak absolutely to this model. But again, it's a model, and there are clearly a lot of effects going on. And as I've shown as well, so what resting state may be something very different from when you are challenged with a specific task or specific stimuli, right? So I don't think that the model can explain everything and there will be, the whole unified theory of psychedelics, but I do think it might indeed explain some of the effects, yes.
Thank you. That was very enlightening.
Yeah, you can just... Okay, so
For the next
thank you so much, Katrin. So we are now done with you. Thank you again. I think we can just jump in.